At its very core, drug science must remain independent. This means we don't accept sponsorships. It's with the support of the drug science community we're able to do this and make the podcast in the first place. If you're able to become a drug science community member and support the show, you too will be supporting the dissemination of evidence-based drug policies. Without you, none of this would be possible. For anybody interested, there's a link in the show notes. Thank you. Hello and welcome to the Drug Science Podcast with me, David Nutt. Here we're bringing together experts and activists for a rational, honest and informed conversation about drugs.
Welcome to the Drug Science Podcast with me, Jo Neal, and Hannah Thurger. Today we have a very special guest, Professor Mittal Mehta. And I have known Mittal for some time, a lot of it through our interaction with the BAP, the British Association for Psychopharmacology. Mittal started out his career with a neuroscience degree at the University of Cambridge. He then went on to do his PhD on the neurotransmitter dopamine and its role in memory and learning. Then, once he'd finished that, he went to Imperial University in London, where he started a long career in neuroimaging. And this is one of the topics of today's podcast. Hannah and I hope you listeners, are really interested to know really what is neuroimaging, how does it work, what is it used for, and how has it kind of advanced our understanding of how drugs work in the brain, which I think is what Mittal's been doing, and enabling us to develop better treatments for psychiatry and other illnesses. So he has a research group at the moment, and that uses imaging and behavior for drug development in psychiatry. So that's kind of one of our topics today. He, like I have done over the course of my career, have worked with a lot of pharmaceutical companies, and that's been has been a real adventure for me. Some of the scientists have just been fantastic working there. He's very busy, of course. He's a prof at one of the biggest UK universities. He's now at the Institute of Psychiatry. It's changed its name. Mittal will tell us exactly what it's called now. So he holds many academic roles, and we will cover some of those. He's a research integrity champion. I think that's a really important issue in research. Be very interesting to hear how he's, through that role, making things better. So over to you, Mittal. I thought we could start maybe with your early career, maybe a bit about your upbringing, your school life, and why you chose to devote your life, really, in your career to studying the brain.
Thank you for that introduction, Jo. It's interesting to hear about oneself from someone else's mouth. So it's an unusual experience. So, well, I don't really talk about my early life very much. There's nothing wrong with it. It was wonderful. My parents came over from India in the 60s, and my dad came over first, and he knew someone in Bolton. And he arrived in Bolton, and he said the first thought he had was that he doesn't want to live in Bolton. So
apologies for people from Bolton.
This is obviously Bolton in the late 60s. I'm sure Bolton in the 21st century is a transformed place, and my dad would welcome it with open arms. But he knew someone else in southwest London, in Kingston, and he went there and he thought, this is more like me. Of course, he arrived in the autumn, so of course weather was a big issue coming from India.
Oh, so cold, grey and damp.
It was so cold. Anyway, so I'm very grateful that they chose to come over to this country because I enjoy living here. I love it. And went to school in Kingston. And I think it was quite early on, I thought, I'm quite interested in science. This seems like an area that I can understand to a degree. I felt like I could navigate it. I did okay at school. And with a few friends, we all joined the British Association for Young Scientists, and we invited speakers. It all sounds very serious, but I think we were making it up as we go along, and that's generally my rule for life.
Yeah.
If you make it up as you go along,
Yeah.
And, there'll be some good decisions in there, and you'll do things you didn't expect to do. So that was good. My friend's dad was the producer of like BBC science programs. So we got to go to the BBC studios and watch like the pre-recordings of programs like QED and Tomorrow's World. That was all very exciting, actually.
Great fun little.
Yeah, it was great. It was really good. And so it was very clear to me that I had to apply to do science at university. I thought I was going to be a chemist, and I was incredibly wrong. Because I thought it was awfully dull at university. But I was very lucky to get into Cambridge that have the tripos system, so you can change. So I switched to the biological sciences, which I really had no experience of. I was studying things like materials science and physical chemistry. And I thought, neuroscience is for me. So I had a bit of a standing start in neuroscience during the second year of university, which was quite hard work. But, a lot of people do hard work, so that's fine. And very fortunate that Cambridge had decided to instigate a neuroscience final year course. And I did the second year of that. But I met my future PhD supervisor during my second year at university.
Who was that?
That's Barbara Sahaki.
Oh, I didn't know it was Barbara. Right, fantastic. So she's one of the best known neuropsychologists, I suppose?
Yeah, she was doing neuropsychology and psychopharmacology in humans. She'd done a lot of work on the cholinergic system, and in the group they were currently studying the noradrenergic system. And so I thought we should study the dopaminergic system. It seemed like the natural progression. But I was fortunate in getting a little scholarship from the university to do some summer work as a scientist. And the study that Barbara was running at the time with Rebecca Elliott, who's a professor in Manchester, actually now, was to look at the neuropsychological deficits in people who were recently diagnosed as HIV positive, but were asymptomatic in terms of symptoms. And we were able to show that there were some mild cognitive deficits in executive function and episodic memory in this group of people. So they very kindly put me on the paper as well. So I got, very interested in the power of neuropsychology to pick up deficits that weren't obvious when you were speaking to someone. When you actually break down the task and take it out of context, you can be sensitive to a deficit which may not come across in your everyday interactions with people. So the opposite, I suppose, of what you'd call ecological validity. I thought that was fascinating. And then I actually went and worked for a charity briefly after university. And it's through that charity that I met my current wife, my only wife I've ever had, actually. It was also my current wife.
Well done, Mitchell. And I know you have twin boys, actually.
Yeah, twins. Yeah, they've just passed their driving test, so life's a bit scary.
Whoa.
Thankfully, they both passed first time because the tension in the house was palpable in between the two tests.
And they'll both be going to university or onto their, leaving school.
Yeah, that's exactly what life is full of now. Yeah. So they definitely don't want to come to King's. That's what they said. They fear bumping into their father at King's.
Yeah, my daughter would not have come to Manchester. Absolutely not.
No, it's amazing, isn't it? You think I'd be promoting King's for them, but no, it has the opposite effect, of course. And, they can do what they want. I'll be very happy if they're happy, of course. Yeah, so that was fun, working for that charity. But then I realised actually I wanted to work in, I needed to look for a job and think about my career and things like this. So Barbara very kindly got in touch because they had a job going, an RA job going, a research assistant job going, actually, doing more neuropsychology, this time in dementia. And I jumped at that chance. Marvellous. And it went very well. But Barbara was very kind. And she said, look, if, you've done the tests and the follow-ups, assessments, etc., and you've got time, you can run your own project. And so with some other people in the lab, we came up with this protocol to test people who'd had brain surgery or stroke to try and understand sort of classical neuropsychological methods we were using to understand the cognitive deficits with people with very localized brain damage it was quite a tough study. I don't think I realised how tough recruitment is at the time. So we didn't get enough participants to make it publishable, but we did get enough participants to give us some insights. And also, I mean, I was very lucky. I learned a lot from doing that study as well. And actually, of course, we'd probably go in with brain imaging now first rather than looking at lesion patients. And that was great. And the first day that I got my own data, so I'd say my own data, of course, from a study where I was involved in the inception and all the approvals and the recruitment and the testing and the analysis. I remember that really well. It was one of my favourite days.
Well, it's a very special time, isn't it? Yeah, the first study that you've designed and seen right the way through to conclusion.
It is. I thought it was amazing, yeah.
Do you, Hannah?
Yeah, no, it's a great feeling. I think I had a similar story of working for a charity after my PhD and then just really missing research and missing getting your own data. So yeah, I can really relate.
So it's wonderful. So now, of course, in my own group, I'm the second or third person to see the data. So,
yeah,
in many ways, there's a loss there. Of course, there's a huge gain, of course, because I get to work with amazing people, a lot of students, wonderful, and don't have to do that daily hard graft of collecting the data.
It is a hard graft.
And you realise, Mittal, you can't do everything when you get more.
No, but I do miss. I mean, I suppose if I had to pick one thing that I miss from not actually running the studies, it's that really. And if I could, but there's lots of other things that I do that are wonderful as well. But that's an amazing feeling. I loved that feeling, and I was hooked, I suppose, as they say.
Yeah. Yeah, from then on to your original research. So then you did go on to do brain imaging.
Yeah, during my PhD, which you'd already said, we're trying to look at dopamine function in humans. Surprisingly, few people had done that until then. This was in the 90s.
Well, I guess dopamine more traditionally associated with reward.
Yeah, I was looking at it in the context of working memory, mainly episodic memory, these sorts of things, executive function.
And I think our listeners will be really keen to learn about neuroimaging, so what is it all about? Actually, I was swimming in the sea this morning and talking to some of the women, and they were saying, what was I going to do today? And I was saying I was interviewing, or we were having a discussion on the podcast about imaging, neuroimaging, and they didn't really know anything about it at all. And these are quite, smart women. So it struck me that there'll be many people listening who really don't know anything much about neuroimaging. So it'd be great for us to learn.
Of course. I was very lucky, though, because during my PhD, Cambridge got a PET scanner, and I ran the first functional neuroimaging study on their PET scanner. In fact, it was also the first psychopharmacology study. We used methylphenidate, which is a treatment for ADHD. And there was some work that suggested that enhancing dopamine function would enhance working memory. And that was all in rodents and non-human primates. Really beautiful work, actually. And very little work in humans. And it's very difficult to boost dopamine function in humans. We don't have loads of good drugs. And the ones that we do hit multiple systems, and methylphenidate's one of them. But it's safe to give to people as a single dose. And so it was.
Well, it's a clinical drug, isn't it? So we know it's safe. And that will just increase dopamine, but all over the body and the brain, I suppose. Everywhere.
Correct. Yeah. So you have to screen people for cardiovascular dysfunction and things like this, because you have to do it safely, of course. It's research. We put them in the scanner, and at that time we used PET imaging to measure brain function.
So what is PET?
So PET, positron emission tomography, you can label pretty much any compound by replacing one of the atoms with a radioactive atom. Okay, so you can replace carbon with a carbon-11, if you got the chemistry right. You can replace an oxygen with oxygen-15. Okay, and these atomic numbers are different to the stable one you'd see in the periodic table. And it makes the compound unstable. And these are unstable in a way that they actually emit a particle called a positron. A positron is the antimatter of an electron. So when this positron comes across an electron, they actually annihilate each other. Okay, they just produce energy, gamma rays. And the gamma rays are released, and the PET camera well, a PET scanner is really a bunch of cameras that pick up gamma rays. And there are millions of these events. And you can put them all together and you can work out where these gamma rays are coming from in the brain. They're coming all over the place, from all over the place. So you can build up a density map. And that tells you where your ligand that you've injected that's got this radioisotope attached is accumulating. Now, you can label water. Water diffuses freely in the blood. And then that tells you where in the brain is there more or less blood flow. And we know from back as early as at least the 50s, if not a bit earlier, that areas of the brain that are more active have a little bit more blood flow going to them. And so we can track that with PET imaging. It's fantastic. And so we did that, and we saw the areas of the brain that were modulated by methylphenidate, and there were exactly the same areas of the brain that are modulated by dopamine and agonists, so drugs that mimic dopamine in the brain, in the non-human primates. So that was fantastic to be able to see that in those brains, of course. And we've moved on from that now, because now you don't have to do PET imaging to look at brain function. Okay, so the limitation of PET imaging is that you're giving people radiation.
And is that quite dangerous, or it's very low level, I'm guessing?
Very low level, but you don't want to give people above a certain amount. There's only so much you can do. So it's tricky for longitudinal studies or repeated studies where you might have multiple doses of drugs, all these sorts of things we'd like to do. So that's one limitation. Another limitation is the temporal resolution. So the amount of time you have to scan to get a data point. So with PET imaging with water, it accumulates the information over 30 seconds. Now, over 30 seconds, a lot can happen in the brain. Okay, and so the temporal resolution is very poor. So you can't really pick up fast events, and you can't do lots of events, and you can't scan lots of times. So it was a fantastic method for looking in the brain, and we still use it. I'll explain what we use it for in a moment, but we switched to fMRI. So after I did my PhD, I actually went off to learn PET imaging in much more detail, but for another reason, which is to measure the brain receptors. So PET is exquisitely good at this. You can label a molecule and it will bind to a specific receptor in the brain, and then you can basically see where that receptor is. You can see its density. You can see drugs binding to it. It's fantastic.
Mitchell, there may be listeners who don't know what a receptor is.
Oh, right, yeah. So a receptor, if you envisage the brain as this huge mass of neurons, okay, these are the cells in the brain. They project, we use the language of trees to describe what they are. So they have it's an arbor, a dendritic arbor, we call it, and arborization. So think of them as like trees with all these different branches coming off, okay? And that's your neuron. It's an incredibly complicated cell, and they want to talk to loads of other cells, okay? But they don't physically connect to these other cells. There's gaps between them, and chemical messengers go across these gaps to communicate between the cells. And you have chemical messengers from cells that communicate very locally with the next cell along, but you also have some cells which project widely across the brain and influence brain activity across multiple brain areas. And drugs like dopamine do that. They can influence multiple areas at the same time. And so they can really influence the state of multiple areas of the brain as well as local activity. So the receptors are like the locks that pick up these messengers that are going across between these tree arbors, if you like. And so we can give drugs which mimic those. And so we can enhance or reduce the effective role of these drugs by giving people their medicines or compounds for research. And so we can measure how much of that drug has got into the brain. A really good way of measuring dose. Sometimes the amount in the brain differs from the rest of the body because the brain has its own way of controlling what's inside it.
Which is a unique problem in drug development for psychiatry, is getting drugs into the brain, isn't it? And then knowing how much is in the brain.
It's a huge problem. And in fact, we're doing a lot of work now to try and solve this because we don't have loads of developments in psychiatry in terms of new drugs. Okay, I wouldn't say there's nothing because things have come through. There are some new drugs, And there are drugs in the pipeline that look promising.
And psychedelics. We just get psychedelics in here.
Yeah, so I was going to say that. Yeah, well, I can mention psychedelics. I mentioned it 3 times now, so maybe one of the psychedelic researchers would appear. So no, it hasn't happened. Okay.
Oh, it will happen.
We must be busy doing another podcast. So, the psychedelics are actually exactly what I was going to come to, because you've got this very poor pipeline. Okay. I have to be careful because there might be something amazing in that pipeline that transforms everything. But it is poorer than we'd like it to be. In the last 10 years, we've got about a dozen new drugs in psychiatry.
Yeah. And they're not so new, are they? That's what always bothered me.
Well, no, they're new. So they're new drugs, okay? And you compare it to another field like neurology, which I suppose is close in that the brain is involved, okay? But they're different disorders. And you've got over 50.
What bothered me about all the time I spent with drug discovery in psychiatry was that lots of the drugs were similar to other drugs that were already on the market. You know, the innovation in psychiatry wasn't as it is in cancer.
Yeah, a lot of those drugs are tweaks of existing drugs. They might be really useful tweaks because they might reduce the side effect, they might increase the absorption, they might have different metabolites. But you're right, in terms of innovation, there was very little.
And why has there been such a lack of innovation in psychiatry specifically? What do you think are some of the key reasons?
Well, a very good question, yeah. Where psychiatry is benefiting is from repurposing, of course. The top 2 examples are ketamine and psilocybin. So ketamine is still used as an anesthetic, developed in the 50s. Psilocybin can be synthesized or derived from some plants, from mushrooms, in fact, of course, as we know. And, repurposing in this general sense, these are compounds that have been around for a while. They have another purpose in society, whether it's recreational use, whether it's a medical use. And then we're trying to bring the evidence to the table that actually this is a treatment or a treatment regimen that could benefit patients in the formal setting of the psychiatry clinic as well. And this is where I think not only repurposing is helpful, but reformulation or rethinking existing drugs to make them better. Innovation, new drugs, very exciting. As a researcher, you want to look at the different mechanism. But actually, as I go through my career, I'm thinking if I'm going to make a difference, let's take what we do know and make it better as well.
Better, and ones that can be integrated into the current healthcare system. I mean, that's the big question right now, is how are we going to get these drugs to be integrated?
Yeah. So I took over as director of one of the centres that we have at the Institute of Psychiatry, Psychology and Neuroscience.
Thank you.
Is the full name of where I work.
I can never remember, Mitchell. Thank you.
Yeah, so just to answer your question at the beginning and start this podcast, I work at the Institute of Psychiatry, Psychology and Neuroscience at King's College London. One of my hats is director of the Centre for Innovative Therapeutics, or CFIT, as we like to call it. And we work very closely with companies. And we want to transform how universities work with companies, particularly, of course, in mental health research, neurology research, to bring new treatments, bring better diagnostics. And, we're not a tech transfer office. We're not like a dating agency. We are scientists. And we know from research in the States that the closer the integration that a company has with scientists, the longer they work together in partnership, the more successful the outcome is likely to be. So we're using this model. So we want to be science-led in our discussions, bring in people as appropriate who actually know what they're talking about, work on protocols, methods. And there's a couple of companies that were really not in psychiatry that are now in psychiatry. We're very proud of that. We've got a licensing agreement for a novel formulation of cannabidiol to try and deal with the issue of bioavailability.
Oh, that would be so useful, Mitchell, because that's a real issue.
The next target we have is to improve clozapine. So it's very exciting. So clozapin's probably the best drug in the treatment of schizophrenia, but it is not first line because it has terrible side effects, and you have to monitor the blood levels, And you have to monitor some of the immune system markers as well, because you can get some catastrophic responses. They're not very common, but of course it's awful when it happens. And you also have terrible side effects like constipation, and people just stop taking the medication, of course, and then that can be quite problematic as well. So if we can improve the brain penetration, of course, in a way that can be provided, that can be scaled up, can be safe, provided to patients, acceptable to psychiatrists, acceptable to patients, then we could transform the current treatments as well. So we could integrate science with current treatments, but we do need to have a bit of muscle behind this sort of work, because someone's got to push the development. Someone's got to be driven to get this out there into the market so patients can benefit from it. If it sits within a research group, it might be amazing, it might be interesting, but that's no good if it's not useful. So that's what we try and do.
Mitchell, can you lose the immune effects in certain people? Can you lose that effect of clozapine?
I don't think so. I think if people's neutrophil count goes down or they show signs of agranulocytosis, you just don't give them clozapine. Yeah, maybe some people can go back. That's a question for my clinical colleagues, but we can work at the scientific level to try and improve this. So we're really excited about the prospects of repurposing. And of course, we've got these lovely examples with the psychedelics and with drugs like ketamine, but we've also got loads of other examples on our doorstep. And it's about selecting the right ones and answering the best questions. So I think, that freedom to study any drug and to work with anyone is something that I really value.
Yeah.
Because we don't know where the answer is going to be.
That is fantastic, Mitchell. Absolutely fantastic. And you're so right about the repurposing. The other thing you mentioned is the long-term relationships with people in the industry. And I found that when I worked with industry, that the longer you worked with people, the much better the outcomes were. Yeah, it was so rewarding. Yeah.
Definitely. Well, it's relationship building, but it's also trust and knowledge. Like any colleague, you get to know them. You get to know, when you want to work with this person, what their strengths are, what their weaknesses are. And as you said earlier, there are a lot of fantastic scientists in companies. Not all these are drug companies, of course. There are different types of companies you might want to work with on the diagnostic side or the digital side as well. There's an awful lot to do.
Yeah, and that's very exciting.
We can help a little bit. We're delighted.
Yeah, that's great, Mitchell. That's so good to be sort of, bringing that in.
Well, I can get to Hannah's question.
Oh, yeah.
I want to repeat it for people with a poor memory like me.
So it's trying to think, what are the specific barriers to innovation in psychiatry that maybe the other fields haven't suffered with so much?
Yeah, sure. Well, there are lots, but let's highlight a couple here that I think are very important. Okay, the first one is we have to recognize that even though the brain is involved in psychiatric disorders, there's a lot more going on, okay? And we know that your socioeconomic status, your immigration status, for example, and your financial status, they can all influence your risk of developing a psychiatric diagnosis. The experiences in early life can have a profound impact, as you grow up, go through adolescence and into adulthood. And drug use, of course, particularly in your younger life, might have knock-on consequences on your risks. And so, simply thinking about psychiatric disorders cross-sectionally, which many people don't, of course, it is going to only get you so far, of course, and that's a complexity. I think the second challenge is, maybe I should be careful what I say here, but anyway, I won't be careful.
No, don't be careful, Mitchell. Don't be careful.
Disorders don't really exist, of course. Okay, so I'll qualify that, of course, so that doesn't become a soundbite. But maybe people won't be using me as a soundbite. But who knows? So, psychiatric disorders don't really exist. What do I mean by that? What I mean by that is the definition of a disorder recognizes challenges that someone might be having recognizes difficulties and problems someone might be having in their lives and highlights specific symptoms people might have that are certainly worthy of treatment, okay, or, looking at some way of improving that. But the idea that, that there is this clean entity called depression or schizophrenia, whatever, because, of course, we know it's been written out a lot how they are defined based on observation alone. They are defined for pragmatic purposes of trials and treatment and what you need to do in order for a clinic to be successful and for you to share your learnings with other people all over the world. So, pragmatically great. Neuroscientifically meaningless, of course. We can look at biases and differences in the brains and the body of people with certain diagnoses, that's fine. But there's an awful lot of overlap. And these are not neuroscientifically derived entities. And so a lot of my colleagues are now working on trying to develop a more biological understanding across disorders. Some people call it biotype, some people call it transdiagnostic research. These are different approaches to the same issue. And so we've known for decades that we probably need to turn this whole thing on its head. But that requires large data, cross-group, cross-country collaboration. And that's happening now. And now the UK are back in Horizon Europe. Yeah.
Hooray.
So we actually, it's appalling really that we are even out of it, even for a nanosecond. But, the idea that we shouldn't have to, we don't want to take part in, or we can't take part in these big collaborations across countries means that the actual research questions we think are important, or some of them we think are important, can't be addressed, or we can't be involved in addressing them. But now we can again, which is fantastic. And these large data sets, this data sharing, which everyone's really, really up for, which is, again, fantastic, has really taken off. And in fact, I've got a fellow in my group who is amazing at working with big data. Okay, something that I understand, but I certainly couldn't do. So, I love it. I love it having a group who are full of people that can do different things than what I do. And she's doing amazing work. We're really trying to understand what the transdiagnostic elements are at the brain level, particularly in neurodegeneration and psychiatric symptoms in neurodegenerative disorders. That's the project we're running at the moment. We're really interested in seeing what we can do for psychiatric symptoms in older people in particular. And that's great. And I can't wait to see what happens there. I mean, time doesn't move fast enough sometimes.
No, completely. I know massive role of team science. I think it's so important to share data. I think that's so important. And you kind of touched there on neurodegeneration. So you at all excited by the potential role of psychedelics in neurodegeneration?
I have no idea what the role of psychedelics are in neurodegeneration. I mean, we'll have to wait and see. I think it's all exciting. I mean, if there's a rationale and the studies can be done and the results look positive and the trials are good, I get excited. You get excited at blue sky ideas as well, I suppose. So let's wait and see. I don't know. Tell me what the rationale is and I'll tell you if I'm excited.
Yeah, so more focused on the neuroplasticity and the role of the potential anti-inflammatory properties of some psychedelics. It's just that's my background. I'm neuroinformation, so I'm always intrigued to hear what people think, especially those who are working with the bigger data sets.
Yeah. Well, I mean, that's great. If we could maintain neuronal health, of course, with psychedelics, that's a fantastic thing. I mean, we've got drugs that are very good at promoting neurogenesis already, but they include things like cocaine, I suppose, and we have to remember that we should be careful not to settle too early on single mechanisms, because you can achieve promotion of those mechanisms through multiple routes. We have to recognize that there's got to be a broader action that is probably at work, and that these single mechanisms are part of it. Because if you focus on, it's a bit of a facetious comment with cocaine, I know, but if you were focused on a single mechanism like neurogenesis or synaptogenesis or synaptic growth, there's loads of drugs you can pick, which we know a lot about. And no one's talking about those drugs, of course. And so there's, we've got to be careful that we don't settle down too early and we're open to the data. Yeah.
Yeah, it's a very good point.
Otherwise, we'll make bad decisions.
And wait to see the data. And Hannah, there are a couple of trials for psychedelics in neurodegenerative disorders. So it'd be really interesting. And stroke, of course, is your expertise.
Yeah, I mean, the good thing about psychedelics, of course, is that we know that they're relatively safe, of course. And that is what's really good about them, is that they're having effects that we're beginning to understand better. You know, this current age of psychedelic research is coupled with our neuroscientific capabilities. And so this is very exciting. Yeah. Yeah,
the likes of Chris Timmerman's work, imaging with psychedelics on board, showing what's going is just extraordinary, I think. It's fascinating.
Yeah. I mean, we're doing a study at the moment, with ketamine. And what we really want to show are the functional consequences of enhanced synaptic function, because it's really hard to measure it.
Yeah. How do you test for it?
So there are methods where you can use based on EEG and MEG using modeling, and you can pull out parameters like synaptic gain from those models, and they might be insightful. They can tell you something. There are methods using positron emission tomography imaging. I should say those other techniques, they measure electrical activity in the brain. You can use techniques like positron emission tomography, so ligand binding techniques. There's a ligand which called UCBJ, which picks up synaptic density by attaching to a protein that is present in synapses. And then there are other techniques which we're trying to bring to the fore with MRI, which are to do with the local architecture and local function. So, for example, if you do have increase in synapses, they might not be functional, okay? But if they are functional, then they'll use a lot of energy. Synapses use about 80% of the energy in the brain parenchyma, in the brain system. Okay, synaptic activity and glutamate cycling. So you should see an increase in metabolism in the brain, and we could pick that up with MRI. So we're excited about those sorts of methods and what they can bring to the table.
A quick explanation for our listeners about MRI. What is MRI?
Oh yeah, so magnetic resonance imaging used to be called nuclear magnetic resonance, but we can take pictures of the brain now. So let's call it imaging. Let's drop the nuclear. Sounds a bit scary.
Yeah.
And so MRI is a technique which uses a strong magnet, although you don't need a super, super strong magnet. But the stronger the magnet, the better the signal to noise. The signal that you get, the stronger the signal you get. So you have this huge magnet, and what that does is it aligns the protons in the brain. There's no harm in doing this. The protons are effectively hydrogen, mostly in the water molecules, okay? And you also have a transmit and receive coil, and they use radio frequencies. So you put this large magnet in a shielded room, what we call a Faraday cage, and no radio signals, no phone signals in that room. And then you can send pulses of radio waves into the brain, and they knock these protons out of their alignment with the magnetic field. And these protons relax back to this background magnetic field. And as they relax back, they release radio frequency energy. And that radio frequency energy varies a bit depending on, for example, the local environment of that protocol, if it's in a bound water or free water or whatever. And we can pick this up with the scanner. And you can then construct images. And you can construct images of grey and white matter. You can construct images related to blood flow and metabolism. You can construct images which are related to brain activity and what's happening in the blood. Because hemoglobin, the blood cell that carries the oxygen around, has iron in it. And the iron is unpaired when there's no oxygen there. Okay, so you've got this positive ion, and it causes a local distortion in the magnetic field. So we can pick this up. We call it the Bold effect, the blood oxygen level dependent effect. And this correlates very nicely with brain activity. So we use MRI Bold to measure brain activity, and we use MRI, we call it T1, T2 weighted imaging to measure brain structure. So we can measure brain structure and function, and we can do it all within 15 minutes if you wanted to do it in 15 minutes. Mostly people are in the scanner for an hour and a half because we're greedy and we want to ask loads of questions. And people are very tolerant with us.
And it's very noisy being in the scanner.
Yeah, it is, but not for much longer. So my physics colleagues here are working with the manufacturers to develop silent sequences. So we've got silent sequences running on the scanner now. And okay, they're for brain structure, they're really good. And for some people that can't tolerate the noise in the scanner, some kids, some people with some certain diagnoses might not be able to tolerate the noise, we can now scan them in relative silence, the same volume as the background, like air conditioning sort of noises, I suppose you'd have in a scanner suite. And we can also do it for functional imaging. The quality of the images is improving every day, and so hopefully that will be out for general use before too long. But that's very exciting because that really transforms what you can do in the scanner. Although we do a lot in the scanner already. We can still play noises and sounds as well. People can hear it, and the background is constant, so it does fade into the background a bit. But it is a bit like standing, doing your research study on the hard shoulder of the M25 or something. So you're going to have some distractions with the noise.
Great stuff.
Yeah, not as windy though. It's not as windy.
No, not as unpleasant. Much more pleasant to be in your lab, I would have thought. So, Mydal, such fascinating work that you're doing and all the new data that's coming out. But I see that you're working on hypnosis as well.
Oh, yeah, that's right. Now, that is partly because I've got twins.
Oh.
Because my twin boys were born, and one of the administration departments said, you've got to meet this guy called Quinton, because he's also had twin boys, and I'm sure that you've got a lot to share. So, a few weeks went by, and eventually, Quinton and I had lunch together, and we talked about, we didn't talk about twins, we talked about the brain, and we talked about dopamine, and we talked about brain function, and we talked about hypnosis. And Quinton was just finishing a study in hypnosis. And I said, I think I've got some ideas about how to analyse this data. And so, we worked on this paper. But the real conversation, to my mind anyway, in my mind, what was going on was I've been giving people drugs to modulate brain function. And there are other ways to modulate brain function acutely, in the moment, reversibly. And one of those ways is hypnosis. But the beautiful thing about hypnosis is you can tell people what brain functions you want to modulate with words. It's amazing.
Really?
And so, yeah. And so we designed this study, we wrote this grant, we got it funded. And the area that Quinton really wants to look at, and the study we designed, was around passivity phenomenon. So these are symptoms that you can get in psychiatric disorders, particularly schizophrenia, where you lose control and ownership of, say, a movement or a thought. So you might feel like you've got thoughts implanted in your head.
Oh, yeah.
You might feel like someone else is controlling your movements. So we thought we could study these experiences in relative isolation using hypnosis. And so we started screening people for the degree of hypnotic suggestibility that they experienced. And about 10% of people experience enough that we would put them in this category of having cognitive suggestibility. So we can make, cognitive suggestions to them. We can suggest them they can hear something that isn't there. They can see something that isn't there. They can feel that someone else is controlling their movement.
Wow.
And so we did an experiment, and the design was based on very similar to our drug studies. It was, it wasn't double blind, of course. We couldn't do that. But it was randomized. We did have control conditions. And we used exactly the same analysis pipelines that we're using in our drug studies. And so the study we did, which was published in 2015 now, it's a while ago now, we were able to show that during automatic writing, this sense that someone else is controlling your writing, we saw changes in brain activity in areas of the brain involved in writing, involved in motor function, and such as the angular gyrus. If you damage the angular gyrus, you get agraphia, such as the putamen, which is part of the motor loop that controls the initiation and smooth motor function, and the cerebellum, which is part of the output structures that control sensory motor function. And if we asked people, if we suggested to people that their thoughts weren't their own in our task, then we saw a reduced activity in the language networks of the brain as well. So that's very exciting.
Amazing.
Yeah, and we've done quite a few studies now using hypnosis, and we're collaborating with people in Stanford using the same task. We developed this writing task that allowed you to separate the thought component of a trial from the expressive component of a trial, and they're using it as well. And it's proved very powerful. We just finished a study in which we suggest auditory verbal hallucinations combined with EEG to see if auditory verbal hallucination experiences look more like listening or inner speech, for example, all these different theories about what it's like. So we can test these. It's an incredibly powerful technique. So people think about hypnosis, I think, as the actual hypnosis, that moment itself, the sort of, I suppose, becoming under control of the hypnotist, or, you know, working with a hypnotist. And that's fine. And you can study hypnosis per se and what is behind hypnosis. And some of our research has been looking at that. We call that sort of intrinsic hypnosis research. But what we're doing is using hypnosis as a tool to study suggestions, to study brain processes, functions, and experiences. And so we call that sort of targeted suggestions research as well. So it's important to split the hypnosis research into two. And I'm talking about targeted suggestions, that is using hypnosis in people who are highly suggestible to study specific processes.
And has it given any specific insights into schizophrenia?
So this is what we'd like to do next, is we'd like to do the comparative studies as well. But it's certainly given insights into these experiences. Well, first of all, we know that someone without schizophrenia who is highly suggestible can share some of these experiences the patients have with schizophrenia. And if we were to, I mean, we haven't specifically looked at the same questionnaires, but in other work, we are looking at the same questionnaires and rewording them so the language is different from the schizophrenia language. But if you ask those same questionnaires, you know, you're going to get ratings in the same sort of range as you'd get in people with schizophrenia. Of course, but these people don't have schizophrenia. These people, you know, they're screened to be healthy as far as we know. So this is amazing. So our brains have the capacity for a huge range of experiences. I mean, we know this. You can give a low dose of a drug and you can transform the experience you have of your local environment, your worldview, etc., etc. And we can do it with words as well.
Yeah, and of course meditation as well, another form of another altered state of consciousness that's being researched with some similarities but also differences to obviously under the psychedelic state as well as hypnosis.
Exactly. So, you know, there are lots of ways of getting to understanding these varieties of brain function. And you know, if you can come up with the right questions, then these are amazing research tools as well. And amazing participants.
And Mikkel, do you think people with psychiatric disorders can be helped with psychosis? With hypnosis, sorry.
Well, hypnosis is already used in addictions, and there's some success, of course. Whether it's not really used outside of this, and I don't think we know how to use it outside of those areas yet. And we have to remember that not everyone is suggestible. Well, actually, most people are suggestible, but not everyone is highly suggestible.
It seemed 10%. Did you say?
About 10%. 90% of people are suggestible. They'll follow at least one suggestion during a hypnosis induction session that we'll use in our research, which is quite a controlled, boring session, really, from the researcher's perspective. Lovely and peaceful, though. But about 10% of people we would call highly suggestible and would be suitable to take part in this research. Or maybe they're the people who are more likely to respond to a therapeutic intervention as well.
Okay, so it could be a predictor as well. Are there any factors or kind of common personality traits that you've seen in your research that make people highly suggestible?
This is a very common question.
Yeah, so interesting.
We get asked, or we get asked. And there are some. You know, if you look, you'll find a paper that shows these two personality traits, for example, correlate with your suggestibility, or, you know, for example, being female is a predictor of being slightly more suggestible. Although actually we did the UK norms study recently. We didn't find that effect was as strong as other studies have found it to be. But all the predictors are poor. Yeah, they're all poor. So I think the simple answer is no. We don't have a reliable, robust predictor of who is more or less suggestible. You just have to get on and do it. So
brilliant. That's fascinating. I know our listeners will be really interested in this. So just before we run out of time, I'm quite conscious of time, you are the Research Integrity Champion.
Oh, yes, I am.
For your university. Can you tell us a little bit about this and a bit about, I was always interested in how difficult it was to get negative results published. I know that's a little bit different, but can you tell us a little bit about that?
Yeah, I mean, research integrity is a key pillar in research culture, which is really about honesty, rigor, transparency, accountability, responsibility, caring about your researchers, all this sort of stuff. It's really important because first of all, you want to publish things that represent the data you've got, okay, that minimize your biases and hopefully can be replicated. But not every robust piece of reporting is necessarily going to be replicated, but it might be robust. You want the ability to replicate to be open to others. So having open protocols, open data where you can. And the responsibility, though, accountability and caring is also a really important pillar as well, because you want to have an environment where you have a positive research culture, where people feel they can come forward. People are happy to raise issues, raise concerns. People are happy to talk about the issues. And so they don't live in fear of the supervisor, or they're not hand-tied by getting a high-impact publication. They're more driven by having a good publication that can be replicated or they can stand by the data for. Okay, so there's a long way to go because we're no way over all of these barriers. Okay, and good quality research integrity is difficult. It requires constantly being up to date on the best methods. It requires being able to share everything that you can and being organized and skilled enough to do those things. And they're difficult, and you need a team to achieve good research integrity. Research integrity is also about calling out poor practices, and they could extend from not reporting all your results. You might not report some results which don't sit so well with the main findings. You might think they're inconclusive and you're not so sure, but you've got to be careful. You've got to tread really carefully there because you don't want to mislead the readers and the field. And it also goes all the way through to fraudulent behaviour, of which we've got examples where people have literally sat down at spreadsheets and started typing out data and got very good publications from that. So it also speaks to our culture of publication and the way we're rewarded and recognised in the field.
Yes.
So I think...
Irish.
Yeah, exactly. And so it sort of goes through everything we are as researchers. And so, my role as Research Integrity Champion, I'm representing our faculty. I link up with the other faculties and we look at policies and procedures. We write proposals. We come up with ideas. And one of the things that I'm really keen on is creating a positive research culture. So we have our research integrity team who can deal with accusations of fraud and people disgruntled about their authorship position or, all these things that we should take very seriously. I get involved in some of that some of the time. But actually, day to day, if we can create a positive research culture, maybe we can reduce the chance of these things happening, but maybe we can improve the quality of the research that comes out. Maybe we can improve the reward and recognition of people that engage in a positive research culture. And so certainly that is something we're aiming to do, is to use research integrity not only to bring forward people who are involved in bad practice, but also bring forward and highlight people involved in good practice as well. And so we're putting together prizes, awards as well for people, and some of the work that people do is fantastic as well. We're working on the policies, we're looking at things that we could do as well to enhance practice for everyone. So we've just got some money to bring together all the different branches of good research integrity practice that we do so we can share best practice. We can enhance what people say that they will do, and hopefully that will also enhance what people will do.
No, it's fantastic. Yeah, it's like a celebration as well, rather than just highlighting the negative aspects, creating an environment where people are celebrated for the good they do.
Yeah, we run an annual research festival, and one of the slots in there is reserved for the winner of the Open Research Award that we give out annually. And that's a very important moment. And it's usually, I find it quite inspiring what some people have achieved in open research.
Absolutely.
Sharing their data, getting people to be open about their ideas and their predictions of their study and writing those down and publishing them beforehand.
Yes.
When you actually run your analyses, you can check it's the one that you wanted to do. And so any additional analyses you can do are then very clearly exploratory. And so then, of course, the challenge here is to prevent people reporting what they found and not reporting the findings they didn't like. It's about reporting the findings that you wanted to test and also reporting findings you do like.
Yeah, and sometimes the findings that you didn't expect, they're the most exciting.
Yeah, exactly. And I always wonder for people outside of core science what they think about integrity and and how we work on our data, what we report, what we choose to report. And, unfortunately, the truth is we're not as systematic as we'd like to be, folks. And we could be better and we could improve practice. Just like in any area of life, you can improve how you go about it.
And people tend to publish their best results. And it's so important to publish the negative findings, the things that didn't work.
It is. Exactly. You don't want people repeating things that are not going to work.
No, that you need an army of people.
Yeah. Giving drugs to people or, studying experimental animals, you really don't want to be repeating studies that you don't have to.
Absolutely not. It's completely unethical.
But also it's about data sharing as well. Even if you've got a negative finding, it might be that your sample size was too small. And when you combine it with everyone else's data in the area, you've got a very clear positive finding.
And there's no excuse to not data share, I don't think. That's so sort of fundamental.
Yeah, I think there are very few examples where you've got small numbers of people with rare disorders and there's a risk of identifying people. And maybe but these are rare examples. I think the norm should be to share and the exception should be to not share.
Yeah, that's lovely, Mittal, actually. That's a really, really great way to put it. So, Mittal, we're nearly at the end. Is there anything you'd like to mention to our listeners we haven't covered?
Of course, I don't have to talk about my own research studies, but, there are some studies that we've done that are in the mix that I'd love to talk about more. We're working on trying to understand psychosis as it appears sometimes in Parkinson's disease, for example. And we're trying to look at novel ways of reducing hallucinations. About half of patients with Parkinson's disease, which is a movement disorder, or at least it's diagnosed as a movement disorder, but we know it's a multi-system disorder in the brain. About half the people, if you follow them up long enough, will develop psychosis. So they'll have hallucinations mainly, some will have delusions, and it can be quite distressing for the patients and their carers. But there's really very little in terms of treatment options for those patients. Very little understanding of the neurobiology. So we're working on that, which is very exciting. We're doing a lot of work in mood disorders, trying to understand how we can assess novel treatments better. There are disorders such as bipolar depression, which is people with bipolar disorder who don't really experience full-blown mania. They might have hypomania. And there's really no novel treatments for this group as well. So we're working in these areas trying to develop better imaging markers so we can then have a method. First of all, we want to understand disorders better, but then we have a method to assess new treatments as well. And so we're writing up some research in those areas that we're really excited about.
Brilliant. Well, maybe we need another podcast with you to talk about all these research projects, because those are very innovative, important and exciting.
Yeah. And we didn't talk about the British Association for Psychopharmacology.
No.
which is the association that brought Joe and I together. So, and we didn't talk about so many other things in research in general, I suppose, to do with the culture of research and how we go about research, the challenges in research.
One of which is money. You can't do research without proper money, and that's a difficulty.
Exactly. And the role of government as well. We can talk about that as well. how they set agendas and they determine funding. And it's a real influence. And actually, so, I know nothing about policy, but I recognise that actually to make a difference, you've got to be thinking about policy, process, funding, culture, as well as the actual research questions, because none of it's in a vacuum.
Yeah, that's a very, very good point, actually. And how difficult it can be in smaller institutions. Where the three of us are very fortunate to be in large, well-funded institutions, but there are many across the country that are pretty small and don't have the facilities and the funding to do a lot of imaging research, to use more complex tools and techniques. And I think really this is where all the collaboration comes in.
Oh, yeah, and the open data.
Yeah.
I mean, there are quite a lot of groups in the world who don't have access to imaging.
Oh, many.
But are world-renowned for their imaging research because of the ability to share, to take in data that other people have worked hard to collect. So, yeah. And other things. Yeah, I think we've talked about enough.
Good. Me too.
Yeah.
Oh, it's been a masterclass on neuroimaging. Absolutely fantastic.
Absolutely fantastic. Yeah, it's been a real pleasure talking to you today, Mette. Thank you so much for taking time off from all your work to talk to us.
I'm going to go and fill in some forms now.
Oh, one of the less fun aspects of academia.
Exactly. This is what my children think my job is, talking to other people and filling in forms. They're not that wrong.
Well, there's a lot of that involved, sadly.
Yeah, but there is a lot of that involved. But thank you very much for inviting me. It's been a real pleasure. It really has been lovely to chat to you both.
Oh, thank you.
Thank you, Mette.